Good afternoon. My name is Dr. Nirenberg, and I'm going to tell you about how I deciphered the genetic code in the 1960s, along with two other scientists. Up until this point, all that geneticists knew about turning mRNA into proteins is that nucleotides were coded in sets of 3. But other than that, a lot was unknown. So for my first experiment, I worked with Dr. Matei. This is our little representation of him right here. And so for this experiment, we took a sequence of U's, so our U, our poly U, and we added it to a mixture with cell extract, which is basically all of the components of a cell without the membrane. And to this, we added all 20 amino acids. So here's an example of tyrosine and amino acid. And we repeated this 20 times. So in each mixture, we had 20 amino acids, and one of them was radioactive. All right, from this mixture, we looked to see if protein was formed, and a protein was finally formed when the amino acid phenylalanine was used. This showed that our sequence of U's coded for phenylalanine, and from this we deduced that the protein was made up of the amino acid, and that the codon was what coded for this protein. So we repeated this experiment using poly C's, poly A's, and poly G's. And from this we found that AAA coded for lysine, that CCC coded for proline. Here's an example. And the poly G never worked, so never coded for anything. So we were able to identify 3 different amino acids. So for my next experiment, I worked with Dr. Leader. And here he is, right here. And for this experiment, we took short sequences of mRNA with every single possibility of codons, so every single sequence we could make. So we added this to the mixture, and then we added a ribosome and a tRNA that was attached to an amino acid. So we added this all to the mixture and allowed them to bond together. From there, we used a filter that caught all the ribosomes and RNAs that were successfully bonded to the codons. So all the parts that were bonded and successfully created a protein were caught in the filter, and these were able to be analyzed and looked at. So through this experiment, we identified 61 different codons, and this was a breakthrough that allowed geneticists to create a universal coding dictionary. So here's our universal coding dictionary, and it shows which proteins or which codons code for which protein. These experiments were pivotal in the genetics field.